Welcome back from the break. We're on the home stretch now, right into session 8, global collaboration to support efficient generic product development and regulatory assessment. We do have a slight change on the agenda of presenters with Dr. Sarah Ibrahim presenting first. Dr. Ibrahim is Associate Director for Global Affairs and OGD. Our next presenter is Dr. Nullifer Tample, Associate Director for Scientific Quality, Office of Bioequivalence and OGD, and we're welcoming back Dr. Lei Zhan to present on FDA EMA Parallel Scientific Advice Pilot Program for complex generic and hybrid drug products. And finally, we welcome Brian Folian, Deputy Director, Office of Study Integrity and Surveillance in the Office of Translational Science for CDER. Please join me to welcome our first presenter, Dr. Ibrahim.
Thank you for the kind introduction. I'm Sarah Ibrahim, the Associate Director for Stakeholder and Global Engagement in the Office of Generic Drugs, Center for Drug Evaluation and Research at the US FDA. And today I'm delighted to share with you experience from the generic drug cluster. In 2022, the worldwide market for generic drugs achieved a value of $343 billion. Projections indicate that this market will grow to reach $460 billion by 2028, with a compound annual growth rate of 5% during the period from 2022 to 2028. This growth is largely driven by an aging population and an increase in chronic diseases. Now, in an effort to manage healthcare costs, governments in developed nations are actively promoting the use of generic drugs. These medications provide the same therapeutic benefits as their brand name counterparts, but at a significantly reduced cost. Meanwhile, in developing countries, generic drugs offer a potential solution to the challenges of affordability and accessibility in the healthcare sector. By providing cost-effective alternatives to brand name medications, generics can help overcome financial barriers and improve access to essential treatments. In the past, the United States was the primary source of medication production. However, over time, the manufacturing of drugs, especially active pharmaceutical ingredients, has shifted overseas. The cost benefits offered by countries outside the U.S., such as lower labor costs, cannot be matched by traditional pharmaceutical manufacturing technology in the U.S. Now, to illustrate how global the generic drug supply chain has become, we analyzed the location of API manufacturers for marketed ANDAs with active and pending manufacturers. From 2012 onwards, there has been a steady increase in the percentage of ANDAs where an API manufacturer is based in India. Conversely, the percentage of ANDAs where an API manufacturer is located in the United States has been on a decline since 2011, with only a few exceptions. Despite the global shift in drug manufacturing, the United States continues to hold the largest portion of ANDA finished drug formulation manufacturers for marketed ANDAs. However, there has been a consistent increase since 2012 in the percentage of ANDAs where finished drug formulation manufacturers are based in India. On the other hand, the share of ANDAs where the finished drug formulation manufacturer is located in the United States has also been decreasing since 2011. Now, given the global and complex nature of our generic drug supply chain, the FDA's international presence and engagement play a crucial role in securing our supply chain. The FDA's regulatory system ensures the quality and safety of drugs produced globally, even before these products hit the market. The FDA stands by the rigorous standards and regulatory inspections that ensure the safety and quality of all generic drugs, affirming that they are just as safe and effective as their branded counterparts. The Generic Drug Global Affairs Program is an initiative that aims to strengthen the FDA's global collaborations. Effective communication with stakeholders is a cornerstone of this program. By maintaining open lines of communication ensures that all regulatory parties are informed about regulatory requirements, changes in policies, and emerging trends and challenges. The program also focuses on identifying opportunities for early regulatory alignment. This involves working closely with international regulatory agencies to discuss standards and procedures for generic drug approval under development. Such alignments can pave the path for harmonization that may expedite the approval process, reduce duplication of efforts, and ensure consistent quality of generic drugs across different markets. Proactive engagement with regulatory counterparts is another key aspect of the Generic Drug Global Affairs Program. The FDA actively collaborates with other regulatory agencies worldwide to share knowledge, exchange best practices, and coordinate efforts. The Global Affairs Program has a number of engagements and initiatives focused on international dialogue that promotes consistency, bringing efficiency in the harmonization process. Strategically taking a collective approach to establish more effective, consistent channels of information sharing and communication. In its inaugural year, the Office of Generic Drugs established the Generic Drug Cluster, a critical initiative aimed at fostering global partnership with leading regulatory agencies worldwide. This initiative is designed to address global challenges and opportunities in the generic drug industry. The Global Affairs Program focuses on establishing targeted outreach to support FDA regulatory needs, involving communicating effectively with global stakeholders. In addition to targeted outreach, the program also emphasizes on regulatory strengthening and capacity building of regulatory systems. This involves providing technical assistance to regulatory agencies worldwide, with the aim of enhancing their ability to oversee the safety, efficacy, and quality of generic drugs. The harmonization of regulations plays a crucial role in the global supply chain of generic drugs, as it helps to eliminate redundancies and inefficiencies. By aligning regulations and standards across different countries, we can ensure that generic drugs are manufactured to consistent quality and safety standards. This not only protects patients from substandard or counterfeit drugs, but also reduces the time and cost associated with bringing new generic drugs to market, thereby increasing patient access to affordable medications. Harmonized regulations enhance transparency and traceability in the supply chain for generic drugs. This improvement aids in monitoring and reporting adverse events related to generic drugs, allowing for early identification of potential safety concerns and the implementation of appropriate measures to safeguard patients. The intricacy of global harmonization is often attributed to the variety of legal systems, political values, and economic structures that exist across different countries and regions. Achieving alignment in regulations and policies often necessitates substantial efforts to reconcile these differences and balance various interests. The formal ICH procedure is a structured process comprising of 5 steps, ranging from consensus building, which involves regulatory consultation, discussion, and finalization, to committee adoption and regulatory implementation. Now, the journey towards global harmonization commences once regulations have been established. This rigorous process involves assessing regulatory differences, conducting scientific and technical negotiations, and bridging the gap between existing regulatory requirements and expectations. The Office of Generic Drugs Global Affairs acknowledges that prospective harmonization efforts, particularly for complex generics, can strategically bridge our regulations while they're being established. Now, global harmonization is a multifaceted and an ongoing process that requires cooperation among various stakeholders. The Generic Drug Cluster serves as a unique platform for maintaining continuous dialogue by fostering a shared vision and promoting discussions as regulations are being established. Now, as I mentioned in its first year, the Global Affairs Program launched two significant initiatives, the Generic Drug Cluster and the Parallel Scientific Advice. I'd like to use this opportunity to not only update you on these programs, but also share the insights gained from both. Globalization profoundly changed the pharmaceutical sector, creating unique regulatory challenges that require successful collaboration and continuous dialogue. Regulators agree in principle of the importance of global harmonization of pharmaceutical regulations, aligning divergent regulations that create hurdles that can be mitigated by leveraging ongoing dialogue and collaboration, where harmonization could also take the form of a slower convergence, with regulatory bodies gradually aligning their standards and requirements with the vision of regulatory capacity building of the agencies that will be responsible for implementing the harmonized regulations. Achieving harmonization of pharmaceutical regulations requires a collaborative effort among stakeholders, convergence of best practices, regulatory capacity building, effective implementation, and an ongoing monitoring and evaluation. Addressing the complexities involved in the development of complex generic drug products requires a collaborative approach among industry stakeholders, academic institutions, and regulatory authorities. The creation of a robust regulatory framework that facilitates market access while ensuring patient safety is contingent upon dedicated research and investment. Such advancement in research could potentially enable generic drug product applicants to rely more on alternative methods, thereby minimizing unnecessary exposure of human subjects. This can be achieved through the extensive exchange of scientific information, streamlining regulatory requirements, and clarifying divergent regulations with our regulatory global partners. Providing developers and manufacturers with the necessary channels can expedite product development and enhance patient access to essential medications. Now, as an agency, we place great value on global collaboration. We acknowledge that for certain products, such as complex generics, that undergo a more intricate development process, access to multiple markets can incentivize companies to proceed with a development program. Now, one of the key benefits of the cluster that has been identified in its role in facilitating a global response, and we actively monitor and analyze emerging trends and developments on the global stage that could potentially impact our regulatory agencies and its policies. This allows us to review existing or new policies to address any gaps, challenges, opportunities, and risks posed by these global changes. We collectively discuss and assess the impacts and implications of these global changes, and the process may involve scenario analysis, risk assessment to ensure we're prepared for various outcomes, ensuring no disruption to medication access to patients. We evaluate and report on the performance and outcomes of our policies in relation to the goals and objectives of our regulatory agencies, as well as the global changes. These ongoing evaluations ensure that our strategies remain effective and responsive to the ever-evolving global landscape. With a globalized generic drug supply chain, global events, international commitments, and policies that arise have repercussions on the drug supply chain and can differ based on the nature, intensity, duration, as well as the extent of event or change. However, these impacts can have drug shortage consequences that affect the availability or accessibility of drugs for patients who require them. Having a quality or safety or efficacy concerns could potentially lead to adverse events and recalls. Compliance challenges that arise in adhering to these regulatory requirements or standards of different countries or regions involved in the drug supply chain. Global changes in policies can significantly impact the regulations of generic drugs, affecting aspects such as innovation and development. With responsive changes in regulatory policies can either encourage or hinder innovation, impacting the development process due to the possibility of arising divergent regulatory responses from our jurisdictions. thereby affecting access to generics globally. As I mentioned, the FDA has established a multi-country forum, or the Generic Drug Cluster, to foster a shared understanding of generic drug regulatory requirements amongst what is currently 7 regulatory agencies and to promote scientific alignment. The Generic Drug Cluster serves as a platform for the world's leading regulatory agencies to facilitate harmonization through early communication and collaboration related to generic drug development. The cluster aims to establish proactive alignment on specific proposed generic drug product review challenges or broader drug class issues through open communication. The cluster meetings have been instrumental in comparing and evaluating regulatory approaches, assessing similarities, and identifying differences. These regular discussions include current review challenges and have led to the resolution of several regulatory obstacles to approvability. Discussions also extend to regulations and guidances under development with regular updates on shared various topics. Now, one significant outcome of these discussions has been progress made in model integrated bioequivalence for long-acting injectable products. Agencies have also committed to sharing data integrity concerns at early stages, allowing for collective awareness of ongoing investigations within respective agencies. Another crucial aspect of the generic drug cluster is its role in anticipating and collectively discussing approaches and challenges due to emerging global regulatory challenges. These could be attributed to global and environmental policy changes and their impact on the generic drug supply chain, prompting the identification of and coordination of multilateral responses to emerging global issues. The Generic Drug Cluster, as I mentioned, is a unique forum underpinned by the confidentiality commitments that promotes the leveraging of collective resources among regulatory agencies. This includes sharing knowledge, expertise, and experiences. The cluster enables our agencies to transition from being data-rich to fostering shared knowledge that enhances our individual capabilities in both depth and breadth. While the cluster typically hosts quarterly meetings, the need for more frequent communications had led to a significant increase in ad hoc meetings. And in these meetings, we have recurring discussions that are held to compare and evaluate regulatory approaches, expectations, similarities, and differences. The cluster participants also share data on human testing of certain drugs where discrepancies exist in the testing population. The assessment of that combined data could support converging recommended testing populations. So if no statistically significant difference is found between patient and healthy volunteer populations, it opens up the possibility for a wider pool of testing participants for these classes of drugs. Now, we published a one-year cluster accomplishments and contributions, and over the past year that represents the cluster's second year, we continue to make significant accomplishments and contributions, and we continue to build upon these efforts. Some of the topics that we have been able to discuss include evaluating the sameness of active pharmaceutical ingredients, particularly focusing on peptide functionality using in vitro and in vivo assays, ensuring that the APIs in generic drugs are equivalent to those in their brand name counterparts. We also continue to discuss complex products that currently have no generic competition, a critical issue that impacts the availability, access, and affordability of these drugs for patients. We have also been working to understand other generic cluster member agencies' approval standards and rationales behind them, as well as their review experiences. This includes specific discussions on partial AUC recommendations for certain products. Member agencies also share their perspective and experience with use of foreign comparator. We also compared narrow therapeutic index classification and bioequivalence approaches among different agencies, discussing challenges with assessing data from alcohol dose dumping studies, as well as share safety data where we compared and investigated bioequivalence studies conducted in healthy subjects versus patient population, especially for complex products where bioequivalence subject variation exists among our agencies. We also conducted investigations on the pharmacokinetic profiles of parenteral nanotechnology products, amongst a number of other recurring topics. Regulatory harmonization and aligning regulations and standards across different countries or regions create a more efficient and effective global supply chain for medications. Regulatory authorities must have the necessary competencies, capacity, resources, and scientific knowledge to deliver their harmonization mandates effectively. And the goal of regulatory strengthening is to improve the ability of regulatory authorities to ensure the safety, quality, and effectiveness of generic drugs. Targeted outreach plays a crucial role in reducing the regulatory burden on the Office of Generic Drugs and ensuring a more efficient review process. It involves communicating effectively with regional stakeholders, helping clarify regulatory policies and procedures that are responsive, targeted, and can help streamline the review process by clarifying regulatory requirements and expectations upfront, helping reduce the number of review cycles for a generic drug application to be approved, saving time and resources for both OGD as well as patients, fostering a more collaborative relationship between OGD and regional stakeholders can enhance transparency in the regulatory process. And our first question, the generic drug cluster 1 consists of 7 regulatory agencies. 2 is a confidential forum for information and knowledge sharing. 3 allows for a dialogue that builds a more robust, coherent regulatory infrastructure for generic drugs globally. Or 4, all of the above. And the correct answer is all of the above. Challenge question number 2. Cluster members focus the discussion on 1. Guidances under development. 2. Agency actions to address global supply chain and development challenges. 3. Current and past ANDA review challenges. Or 4. All of the above. And the correct answer is all of the above. On behalf of OGD, thank you.
Thank you for the introduction. Good afternoon. It's my pleasure to join this webinar to talk about the first harmonized bioequivalence guideline for immediate release solid oral dosage forms, M13A, from an implementation perspective. The ICH Expert Working Group is currently working to finalize the draft M13A guideline. Simultaneously, the FDA is also proactively considering measures to ensure the smooth implementation when the guideline is finalized. And we anticipate that the industry would also be eager for the FDA to implement the guideline without long delays. In that regards, FDA has started to identify some topic areas that would benefit from a proactive approach, and I'll talk about them. I'll also share the ongoing research at the FDA that will help to translate some of the more challenging recommendations in the guideline into meaningful regulatory approaches. The learning objectives are to provide an overview of the M13 guideline series and the progress made today, discuss the next steps by the Expert Working Group, the EWG, for advancing M13A towards adoption by the ICH Assembly, and to discuss the strategic planning for future implementation of M13A. That includes understanding the research areas of interest to the FDA to support the regulatory decisions. Now, EWG is developing the M13 guideline as a 3-part series, each with their respective scopes. M13A is the first guideline in the series to harmonize the scientific and technical aspects of the study design and data analysis to support bioequivalence of IR products. 13B, the next guideline in the series, will harmonize the bioequivalence requirements for additional strengths of a product line, including the biowaivers of additional strengths. And then 13C will cover replicate study design and other complicated data analysis with specific focus on highly variable drugs, narrow therapeutic index drugs, and other complex BE study design and data analysis, for example, the adaptive study design. Now, the three series of the guideline, they are planned for development in a sequential order, following the ICH process for harmonization. So here, the 5 steps of the process are shown here, starting with drafting of the technical document and going up to the implementation of the final guideline. So let's look at the progress of the M13 series. 13A is currently at step 3 of the process, which is the regulatory consultation period. This guideline was subjected to public consultation by the member parties in the first quarter of the year, and the comment periods for all the members have closed, and the expert working group is currently working through those comments. 13B, it is currently at step 1 of the process, which means the technical document is under development. And the 13B expert working group is working towards building consensus on different aspects of the guideline through rigorous scientific discussions. And then 13C, it will start after the 13B reaches step 2, when the draft is endorsed by the assembly. Now let's see how the EWG is working to finalize the guideline for adoption, which is the step 4 of the ICH process. First, the expert working group members will be reviewing and addressing the comments through consensus, and they'll be making necessary revisions and edits to the guideline to support the finalization. The pie chart here provides an overview of the comments that were received for the 5 sections of the guideline. In all, there were 789 unique comments we received, and about 77% of those comments are for section 2. That's not surprising, since the crux of the guideline is in this section that includes study design and data analysis for establishing bioequivalence. The next highest number of comments, about 10%, are on the specific topics which are covered in section 3. In addition, the working group is considering to develop a Q&A document after the guideline is finalized. We acknowledge that the guideline provides broad strokes on certain aspects. Therefore, a Q&A document to clarify the scientific thinking and the rationale, and perhaps using some examples to further explain certain recommendations, can be helpful. Other item under consideration is developing the training materials to facilitate the regional implementation and ensuring consistency for the member regions. I'd like to share that we also received a couple of positive comments, like the one that I'm showing here. Harmonizing on different scientific viewpoints takes a lot of time and efforts. And on behalf of the expert working group, I'd like to take this opportunity to thank you for the support. So coming to implementation, once a guideline is adopted, the ICH expectation is for the regulators to implement the guideline within 6 months. So anticipating the needs and preparing for the implementation is necessary to avoid the delays. Here are some of the tasks that FDA anticipates will need to be addressed. First is developing the training materials. And that includes internal training for our assessors and also training for external stakeholders. Updating the relevant PSGs to align them with the recommendations in the M13A. We also have to update the general bioequivalence PK endpoint guidance for ANDAs. This guidance currently includes recommendations for both IR and MR products. Then we would need to assess if the BE summary tables need to be updated so that they align with the recommendations in the document section of the guideline. And then lastly, conducting research on select topics so we can apply good science in translating some of the more challenging recommendations into meaningful regulatory approaches and decision making. In the next few slides, I'll cover the ongoing research at the FDA. Our research focuses mainly on topics that present either significant change from the current FDA practice, such as the fasting and fed study, or where the agency has limited experience, for example, demonstrating bioequivalence in presence of the acid-modifying agents, which is a recommendation in Section 3.4 of the guideline under the pH dependency, or utilizing the modeling and simulation approach to support the deviations from the recommendations in the guideline. All this research is expected to feed directly and inform our scientific thinking and the regulatory decisions. So first, I'll talk about the bioequivalence under fasting and fed conditions. The draft guideline notes that food can have a differential formulation-dependent impact on the absorption of certain drugs. Applicants should have a thorough understanding of the comparator and the test formulations to determine if testing under fasting and fed is warranted. And the guideline also mentions that the rationale for selection of the study design with regard to fasting and fed may be supported by appropriately validated and qualified either PBPK or semi-mechanistic absorption models. Here I want to share a survey that the generic industry had conducted earlier to evaluate if testing under both fasting and fed conditions is necessary for IR products. The results were shared at the AAM meeting in 2019, and there is a link on the slide for that. And they also shared the results with the ICH EWG. Based on their assessments of the BE study outcome, the industry concluded that 97% of the time, fasting was predictive or more discriminatory than the fed. And only for about 3% of the time, it was not predictive. And that was mainly for low solubility drugs. Now, around the same time, the FDA had also separately performed a retrospective analysis of the ENDAs to look into the BE outcomes under fasting and fed conditions for IR products containing highly soluble drug substances. And what we found was that in some cases, the formulations with excipients such as the swellable disintegrants like the MCC, microcrystalline cellulose, or fillers could affect the rate of absorption of highly soluble drugs in the fed state. So currently, FDA has 2 ongoing research projects for fed BE studies. The first is a grant to develop the PBPK model to predict the impact of food on oral drug absorption of certain poorly soluble drugs. And the second is a contract that was granted to a university in Germany to understand the findings from the FDA's retrospective analysis that I mentioned in the previous slide. This research will be focusing on developing an evaluation algorithm to determine if a fed study can be waived based on the disintegration and dissolution methodologies that can mimic the food-induced viscosity. Now, the results from these research projects will be presented at the public workshop on October 12 of this year. And you can access the details from the link on this slide. We also have internal research projects on the topic of FED-BE studies. So you can tell this topic is of great interest to the FDA. So the first project is evaluating the application of the Bayesian dynamic borrowing approach for FED-PK-BE studies by borrowing the results from the fasting PK studies as informative prior. The second project is investigating how the food-induced increase in the viscosity would impact the disintegration and dissolution of oral IR products. And the results from this project will complement the external contract, which I just mentioned earlier. And the next one is using the PBPK absorption model to evaluate the effect of food on bioequivalence. The work on all this three is ongoing at the FDA, and the results may be shared at a later time, perhaps sometimes next year. The second area of research is the bioequivalence determination under altered gastric conditions. So here, we know that the recommendations for an additional BE study with concomitant treatment of pH-modifying drugs is not explicitly mentioned in most guidance documents from the different regulatory agencies. However, it is noted in the PSGs of few drug products from the FDA and the EMA. For example, FDA's PSG for palbociclib tablets. At this time, the regulators seem to have limited experience and knowledge for the optimum study design for such BE studies. Also, the guideline notes that sponsors may justify not conducting the in vivo studies by assessing the risk of bioequivalence based on totality of the evidence, which could include certain factors which are mentioned in the guideline. So research in this area can definitely better inform the agency about our recommendations. FDA has two research projects in this area. One is an extramural project, which will extend the framework of the PBPK model for FED studies to predict the effect on drug absorption in hypochlorhydric population. The other is an internal FDA research project using modeling and simulation to support bioequivalence study design and evaluate the impact of gastric pH on bioequivalence. This brings me to my last slide. I'll summarize the key takeaways on M13A implementation. 1. We need to remember the draft M13A is an ICH guideline under finalization, and FDA needs to follow the ICH process. So this guideline will not be implemented until it is finalized and adopted by FDA. And that would be the step 5 of the ICH process. 2, FDA has ongoing research to facilitate implementation of some of the key recommendations in the guideline. 3, we intend to update the relevant guidance documents to align with the recommendations in M13A when it is finalized. And then lastly, both the FDA's current practice and draft M13A offer the flexibility of providing appropriate scientific justifications in case prospective applicants want to propose alternate approaches and deviate from the guidance recommendations, including recommendations in the PSGs. This concludes my presentation. Thank you for your attention. I'll be happy to answer questions later in the Q&A session, so please send them in. Before I leave, I have 2 polling questions. The first one, FDA will implement the draft ICH M13A guideline. True or false? This is false. FDA will not implement the guideline until it is finalized. Question 2, ICH M13 guideline is being developed in 3 series. True or false? That is true. I did talk about the 3 series, M13A, B, and C. Thank you again.
Thank you for the introduction. Good afternoon. My name is Lei Zhang. I'm the Deputy Director of the Office of Research and Standards, ORS, in the Office of Generic Drugs at the FDA. Today, I will talk about the FDA EMA Parallel Scientific Advice Pilot Program for complex generic and hybrid drug products. This is one of the initiatives under the FDA Generic Global Program to support efficient generic product development and regulatory assessment. Before my presentation, I would like to have 2 polling questions for the audience. Polling question number 1. Have you heard about the FDA EMA Parallel Scientific Advice Pilot Program for complex generic and hybrid drug products? A. Yes. B. No. C. Not sure. Please take a moment to choose your answer. Polling question number 2. If you are aware of this pilot program, has your company considered participating in this program for complex generic and hybrid drug products? A. Yes. B. No. C. Maybe. D. We do not develop complex generics or hybrid drug products. Please take a moment to choose your answer. This poll will help to understand how many of you may be aware of this program and whether you are considering of participating in the program. Later in the Q&A panel, we will look at the results of these 2 polls. Now let's get back to the presentation. The objectives of my presentation today are to describe the scope of the FDA EMA Parallel Scientific Advice PSA pilot program for complex generic and hybrid drug products, to describe what products are defined as complex products by FDA and as hybrid products by EMA, to delineate the goals of the PSA pilot program, and also to provide highlights of the PSA pilot program and its processes. In addition, I would like to share what FDA has learned through the PSA pilot. Finally, I also will share a few tips for industry to participate in the PSA pilot. The FDA EMA PSA pilot program for complex generics and hybrid products was launched exactly 2 years ago on September 15, 2021. It is an expansion to the existing PSA programs between FDA and EMA for new drugs and vaccines or gene therapies. Similar to the existing PSA programs, this new PSA pilot program allows applicants to engage in concurrent scientific conversation with both agencies on key issues during the development phase, with a focus on complex generic products and hybrid products. The reason that it is a mouthful to say what products are under the scope of this pilot program is because there are differences in how FDA and EMA define and regulate those products under their jurisdictions. The goal of the PSA program is to support global development of these products that are harder to develop as generics or hybrid products under either jurisdiction. FDA's definition of complex products, which was since GDUFA 2, are those products that include complex active pharmaceutical ingredient, complex routes of delivery, complex dosage forms or formulations, complex drug device combinations, and other products where complexity or uncertainty concerning the approval pathway or possible alternative approaches would benefit from early scientific engagement. You can see a few examples of complex products as defined by FDA on this slide. For a full description of what is considered a complex product, you will refer to GDUFA 3 commitment letter or FDA map on the classification of complex products. Complex generic drug products are in general harder to develop with traditional bioequivalence methods, and many of them may require additional studies, including comparative clinical endpoint bioequivalence studies. Complex products in the US have a significant overlap with hybrid products under EMA. Under EMA, complex hybrid products are those whose authorization depends partly on the results of tests on the reference product and partly on new data. For these products, although they considered generics by US FDA, they are not generics under EMA, and they are hybrid products. They are submitted under Market Authorization Application MAA Article 10.3 to EMA, not Article 10.1, which is for generics. On the other hand, some complex generics under FDA's definition could be generics under EMA through MAA Article 10.1 if BE studies are sufficient to support approval. For example, those long-acting injectable products. For the purpose of the PSA pilot program, the products do not have to be both complex generics through FDA and hybrid products through EMA to be eligible. The program is mainly to focus on the products with complexity and can benefit by concurrent scientific discussion with both agencies early in drug development. So what are the main purposes of the PSA pilot? The main goals of this program include to increase dialogue between the two agencies and also use the program to optimize the applicant's global product development by enabling them to discuss specific questions concurrently with both agencies, and also to further provide applicants with a deeper understanding of the basis for regulatory decisions from both agencies. The process can help drive convergency to help applicants avoid redundant replication of work and unnecessary testing replication or unnecessary diverse testing methodologies. And it also will help shorten the time to drug development and approval in both regions or globally. The PSA General Principles document that can be accessed through the link shown at the bottom of this slide gives some high-level overview of the program and its processes. The agencies conduct PSA meetings under the auspices of the confidentiality arrangement between the European Commission, the EMA, and the FDA. It provides a mechanism for EMA and FDA assessors to concurrently exchange with applicants their views on the scientific issues during the development of complex generic product and hybrid products. Alignment between the agencies can lead to more efficient and linear global development, and this program is voluntary for applicants. What may be a good candidate for the PSA processes? Some example could include those products that may have different recommendations in the PSGs from FDA and the EMA. Then the applicant may use the PSA program to propose a study design that may be acceptable to both regulatory agencies, which may be deviate from either agency's product-specific guidances. And this PSA process could drive convergences so that applicant may not need to conduct multiple studies to satisfy both agencies' requirements. Other example can include that the process can be used to discuss the alternative B approaches and the use of modeling and simulation. After the PSA process, each agency will retain its individual regulatory decision-making regarding drug development and the marketing applications. Both agencies will strive to provide PSA responses that are convergent or explain the differences between the agencies. Any EMA fees applicable for the scientific advice are unaffected by the PSA status. And from the FDA side, PSA meetings are not considered generic drug user fee amendments or GDUFA meetings and are not subject to the performance goals for scheduling and conducting those meetings. The PSA process is designed to align the process and timeline of the pre-ANDA product development meeting at the FDA as much as possible with the process and the timeline mandated by the EMA Scientific Advice Working Party for their scientific advice process. Similar to the FDA's product development meeting, PDAV meeting, PSA has 3 stages. At stage 1, applicants submit one single request for PSA letter or justification letter to both EMA and FDA at the 2 different mailboxes. And at this stage, no full package is needed. This is unlike the pre-ANDA meeting package. It takes 14 days for EMA and FDA to decide if accept the meeting request. FDA and the EMA will coordinate and communicate on their decisions. If both agencies agree to accept the meeting package, then it will move to stage 2, where it takes the most time of this meeting process, including the meeting package preparation and conduct of the meeting. During stage 2, there is an optional step that the applicant can send a draft full meeting package to EMA for validation review before they submit the full meeting package. At this stage, FDA will get a copy but will not review the draft meeting package. And I put a star here as this timeline at this stage varies. It will pretty much depend on the quality of the meeting package and how quickly the applicant can address EMA's comments at the draft meeting package. Once the full meeting package is submitted to both agencies, the clock starts as day 1. And the bilateral meeting between EMA and FDA will occur at day 90, approximate day 90, and also the trilateral meeting, which involves applicant, EMA, and FDA, will occur on day 120. As noted here is that those dates, 90 days or 120 days, are an estimate, and it will be largely determined by the annual EMA Scientific Advice Working Party meeting schedule, which normally occurs in a 1-week window per month, and typically in the non-summer months. So the dates can vary depending on when the full meeting package was submitted to both agencies. After the trilateral meeting is held, the FDA will issue the final meeting minutes and EMA will issue their CHMP final advice letter within 30 days after the trilateral meeting. And both agencies will issue the final communication independently. This slide compares a few key features of the PSA and FDA's PDAV meetings, which most of you are familiar with for your reference. One thing I would like to highlight is that PSA meeting will be conducted as a video conference for 90 minutes, longer than the typical product development meeting, which is for 60 minutes. In addition, there's no in-person option for the PSA meetings because it involves both agencies. And also, as I mentioned earlier, the timeline to conduct a PSA meeting can vary depending on the meeting schedule. We have run this pilot for 2 years now. There are several things that we have learned through the pilot. We did not receive a large number of PSA meeting requests. So far, we have 2 PSA meeting requests that has been granted and have gone through the PSA process. This low number may be because the number of the applicants who submit generic drug applications to the FDA and centralize the EMA process is limited. We think that the PSA program can be the opportunity to expand the number of generic drug applicants that do submit applications to both jurisdictions. We also hear concerns from some of the applicants that the PSA program would require additional testing beyond what would be expected if the applicant sought individual advice from each regulatory agency. However, I would like to say that this has not been the experience with the pilot applications. FDA monitored and evaluated the pilot program, and one feedback received was that this is a learning experience for both regulators and applicants to understand the differences in the process and the meeting expectations from 2 agencies. The process also has been used to help convergence on science to address specific challenges in the program. For example, what scientific bridge and/or data can be used to support the use of a common comparator in one study that can support applications to both agencies. In general, the pilot was implemented as intended, demonstrating the long-term potential. The immediate benefits at this point were more visible to regulators than applicants. One lesson learned was that the designed process can be further clarified and improved to encourage applicant participation. Some recommendations based on the program's preliminary assessment are: we needed to have more procedural clarifications, and also the timeline and expectation of the PSA can be further clarified because they have similarity and the differences between the PSA and the current either FDA or EMA process. And also we need to develop best practices for meeting package preparation and participation and share that with the applicant. All these recommendations and lessons learned have been communicated with the EMA, and also some of the key recommendation has already been implemented to further improve the process along the way. Having shared all these, what we have learned, I also would like to share a few tips for the industry to participate. First, you need to know the process. I hope this presentation can help you understanding better the design of the PSA program, its goal, and its benefit and its process. Second, you need to consider and be aware of the jurisdiction differences that can pose some challenge for your specific drug development program, and understand how the PSA program may help you to reduce the redundancy in your development program as one of the possible benefits. Third, you need to define the purpose in order to optimize the trilateral meeting with FDA and EMA, and also what are the meeting expectations. Finally, it is very important for the applicant to work closely with the project managers along the way from both agencies, as there are many coordination needed in order to navigate and keep the communications across all involved parties, both within and outside of the both agencies. Before I conclude, I have 2 challenge questions for you. Challenge question number 1. The Parallel Scientific Advice PSA meeting is part of GDUFA meetings and will follow GDUFA meeting process and timeline. A. True. B. False. The correct answer is B. False. PSA meetings is not part of the GDUFA meetings, and it will not follow GDUFA meeting process and timeline, although we did try to align with the pre-ENDA meeting process as much as possible. Challenge question number 2. What are the following products that may be eligible for the PSA pilot? Select all that apply. A. Long-acting injectable products. B. Immediate release oral products. C. Modified release oral products. D. Orally inhaled products. The correct answer is A and D. Long-acting injectable products and orally inhaled products may be eligible for the PSA pilot. You may refer to my presentation earlier that explains what are considered complex generics. I would like to conclude with 3 key take-home messages. First, ENDA applicants can gain an understanding of both agencies' recommendations on specific questions regarding the global development of complex generic products or hybrid products through the PSA process. Second, the PSA pilot is still ongoing to accept more meeting requests so that areas of improvement can be identified to support future recommendations. Third, we highly encourage applicants to participate in the PSA program by understanding the potential benefit to your specific global drug development program. FDA is mostly interested in submissions that have the potential to reduce duplicative studies for complex generic products that I also provide a few examples at my presentation. Questions about the PSA program can be directed to pre-ENDA-help@fda.hhs.gov mailbox. You can also refer to the PSA and also OGD Global Generic Drug Affairs website as shown on this slide for more information. Thank you for your attention. You may send your questions through the Q&A box. I will be back to address your questions during the Q&A panel later. Thank you.
Hello, my name is Brian Fullen. I'm the Deputy Office Director in the Office of Study Integrity and Surveillance, the Office of Translational Sciences, CDER. Today I'll be providing you a presentation regarding data reliability and integrity for BAB study inspections and global collaborations regarding those inspections. As a disclaimer, the contents of this presentation are my own and should not be construed as expressing the views or policies of the U.S. Food and Drug Administration. The objectives of today's presentation will be to provide an overview of the CDER bioavailability and bioequivalence compliance programs, oversight of testing facilities that conduct BAB studies, as well as provide a high-level and brief description of how international collaboration occurs in the BAB study conduct paradigm. The agenda for today will be to provide an overview of bioresearch monitoring, specifically describe CDER OSIS's BioMo programs, describe how the conduct expectations for BAB studies play out, give you a brief description of how data integrity works in the BAB study arena, as well as a brief case example regarding data integrity for BAB studies, and a description of the complexity and criticality of international collaboration for BAB study oversight, including regard to data integrity. CDER's BioMo program objectives, to start off, the BioMo program is an agency-wide program administered across FDA. In particular for CDER, our primary focus is on ensuring the right safety and welfare of human research subjects, verifying the accuracy and reliability of the clinical and non-clinical trials that are submitted to the agency, and ensuring those studies are conducted in compliance with our regulatory and statutory framework, including ethical treatment of the subjects enrolled in any of those studies. OSIS's BIMA program is effectuated through the Office of Study Integrity and Surveillance, OSIS's vision, and our mission. Our vision in particular is to improve the public health by protecting the study subjects enrolled in clinical BAB studies and promoting the proper conduct of studies under our regulatory oversight. Regarding our specific mission to effectuate our vision, we promote the public health by ensuring the welfare of study subjects enrolled in our trials, as well as verifying the quality, study integrity, and regulatory compliance of bioavailability and bioequivalent studies, non-clinical studies, also known as good laboratory practice studies, and animal studies, AR studies. Here on slide 7, you'll see a high-level overview of CDER, Center of Drug Evaluation and Research's overall structure. Within that, in the orange boxes, you'll see the offices of Study Integrity and Surveillance nestled in there. We have 2 primary divisions within OSIS, one being the Division of New Drug Study Integrity, and the other one being the Division of Generic Drug Study Integrity. Our primary mission, as we just described, is effectuated by supporting our customers and primary stakeholders, one being the Office of Regulatory Affairs, who we work with directly and collaboratively to ensure that we have appropriate inspection oversight of the programs that we oversee, as well as stewarding work with the Office of Compliance, Office of Scientific Investigations, and the Office of the New Drug and Generic Drugs to make sure we are providing information necessary for purposes of application decision making. To effectuate our mission, OSIS has directed our work into 4 discrete domains. The first is we use a risk-based model to ensure that we're appropriately selecting sites that are necessary to inspect or conduct a remote regulatory assessment to ensure we are providing information to allow marketing application determinations by the agency. We evaluate the sites via the inspections or remote regulatory reviews, RRAs, to ensure the study conduct is appropriate and the integrity of those study data. We review the evidence collected via the inspection or RRA and provide reports to the review division regarding the outcomes of those inspections. Those recommendations that OSIS provides are the support function that we operate to inform the review divisions of what is needed, as well as to provide compliance evaluations. In terms of globally BAB study conduct inspections, we go on inspections or RRAs to determine that the studies supporting applications to the agency are appropriately conducted to ensure that the data and subjects enrolled in those data are protected and were adequately and consistently overseen by the testing sites and the sponsors that contracted with those testing sites in most cases to conduct the studies. What is essential there in order to do that is that the records maintained by those sites be sufficient to allow us to confirm each aspect of the study conduct and reconstruct the totality of that. So in order to do that, we review all subject documents, analytical runs from those subjects in terms of the evaluation of the biological samples from clinical BAB studies in order to ensure that the data submitted to the agency is reliable and verifiable. Oftentimes, you hear an interesting phrase bantered about the absence of evidence isn't the evidence of absence. The question always for us in terms of the agency and the BAB program in general is that type of language and thinking relevant or over applied to what we do? As you see, study eligibility in terms of enrolling in subjects requires certain elements of the protocol be demonstrated that a subject has certain conditions or has certain values in their laboratory results. But where an issue is identified, there's some abnormal range, the expectation isn't that silence means that nothing, that the subject must have been deemed to be eligible for enrollment. If there's an abnormal value in that subject's records that raise a question on eligibility, is the expectation for the site and investigator to resolve that discrepant issue. If there isn't a resolution, then you're raising up a study conduct and data integrity issue whereby we can't determine the accuracy of that information. Data integrity here, in the context of, in particular, the BAB program, is that we recognize data is fundamental to what the agency does in order to make regulatory determinations on application provability and other arenas. Data integrity to us is, considered regarding the completeness, consistency, accuracy, and reliability of the information. We look at that throughout the life cycle of the data. It's not just an isolated one-time incident at the end of the study where the study report is produced. There's a design component of that where the sponsor should be determining, considering what data controls are needed to make sure that the records being produced by a testing site conducting the study are complete and consistent and accurate and can be relied on. That should be done throughout the course of the study. It should be concurrent during, and there should be a periodic review. BAB studies are short, so there is an expectation that isn't exhaustive, but there needs to be some consideration. It's a shared responsibility across the sponsor, their contracted entities, the testing sites that conduct the studies, as well as the agency via our compliance and oversight to ensure that we are getting the cleanest, best data to make sure that the products that are being submitted to the agency and the data supporting those products is reliable. I want to provide a quick case example. The study DIRE, which stands for Data Integrity and Reliability example, oddly enough. Here we have a situation where the agency in the BAB program would have conducted an inspection. We'd look at all the documentation regarding the clinical analytical site conduct, look at runs, look at how the subjects were dosed, etc. During the course of the analytical inspection and how they're analyzing the biological samples from the subjects in the clinical, we asked the firm to provide their PK data sets, their pharmacokinetic data sets, and ask them to plot that data so we can look at the PK trends, look at what is going on for purposes of the totality of the population of subjects enrolled in that study. One signal that may have popped out there, right, as you have on the screen on slide 11, you have where subject A period 1 and subject B period 2 seem to be overlapping entirely. One being subject A is on the test and subject B in period 2 is on the reference, right? So how could the test period of subject A and the reference for subject B be mirror images? And then the converse occurs when you look at subject A's period 2, which is the reference, and subject B's period 1, which is the test. You get the comparable mirror images, but you have the reference and test now being reverted, but comparable curves, which raises significant concerns. If you see this once, maybe aberrant, maybe just something that occurs, by chance in a population of subjects. But when you start seeing it more than once across one study, across multiple studies, or within one study, it starts to raise a question on data integrity. And further example here that provided is where you have a population of subjects when the site actually plots out their curves in totality, and the PK data, looking at the TR ratios, the Cmax, you have a distinct separation of populations into two distinct cohorts. It looks like for subjects 1 to 25, they seem to be well below, the TR ratio of 1, but then in the latter cohort, subjects 26 to 50, it's well above. And overall, the study appears to be meeting bioequivalence. So that 90% confidence interval between 80 and 120. What do you do when you see that type of data that's abnormal? The site has no data, no information to indicate how they could have and what they did when they would have observed such a divergent physiological trend. That in and of itself is something that raises significant concerns and questions. If your study records don't allow you to explain the information or how you can have significant overlap between subjects across different studies, or a divergent patient population or subject population, where you have two distinct cohorts within a subject population, how within normal variants would you have two distinct populations. It's unexpected unless there's a significant, examination, right, by the firm to do that. In these cases, that did not happen, which results in what is the outcome, right? What do you have to do? The unexplainable anomalous data to the agency in the BAB context, let me say that, equates to a data integrity issue that undermines our ability to verify that that site is conducting the studies appropriately. The end result there for the agency is we must make a determination regarding the reliability of the studies generated from that site. What is the impact on the submissions to the agency, whether that's pending approved or tentatively approved applications, and whether or not the BAB studies, if pivotal, need to be totally redone. And in the course of this, and overall for these data integrity cases that we're seeing for BABA that we might see, there are significant discussions with the foreign regulatory counterparts, which is the latter part. On slide 15, you'll see a really complicated diagram, and it's intended to indicate that our collaborations for data integrity in the BAB context with our international counterparts is complicated. There are a lot of reasons for that complexity in the international collaboration for the BAB studies that we oversee. In part, we are having significant amount of conversation with our foreign regulatory counterparts. That is without question. Part of the reason for that is that for the BAB studies overseen by OSIS, a significant number of those are conducted outside of the U.S. in whole or in part. We also know that oftentimes the same products are being submitted to other countries for marketing authorizations, and those countries are also having to do comparable inspections of the studies supporting their applications. We know that all regulators, no matter where they're located in the world, do not have unlimited resources. So collaboration to ensure a full, to the extent possible, oversight of the BAB study complex is essential. So there is significant communication with our regulatory counterparts. A key piece here to keep in mind, though, is even with that collaboration and even with the intended comparability, FDA and international regulators' actions in the final setting may differ. We do have similar study conduct expectations. We collaborate in ICH, in ICMRA, in OECD on the GLP side. There's a significant amount of work that OSIS does on a daily basis and weekly, monthly to work with our regulatory counterparts, EMA, MHRA, WHO, Health Canada. But we have to always recognize that our authorizing authorities, our legal frameworks are different, which can lead, even if we're doing comparable inspections and sharing information, to different outcomes, different ability to take actions, whether or not FDA is taking a strong action, EMA is issuing an Article 31, or WHO as a notice of concern. We might in the end have a slightly different external facing result, but that isn't because there isn't comparable support for the other regulatory agencies taking their action, but in certain contexts, the action being able to be proven by any given regulator may differ from each other, which is a key point to keep in mind. So on to the challenge questions. The OSIS BAB inspection program is focused on A, verifying reliability and integrity of BAB study data submitted to FDA, B, reviewing and approving submissions to FDA by verifying the reliability and integrity of BAB study data, C, verifying reliability, integrity, and regulatory compliance of BAB studies submitted to FDA, or D, none of the above. The correct answer here is C. Challenge question, the last one is for comparable inspections, same site and same product. FDA BIMO inspection program actions will mirror the regulatory actions of our foreign regulatory counterparts. True or false? Correct answer is false. And thank you very much. Look forward to talking with everyone shortly.
Thanks so much for all the great presentations in this final session. As a reminder to our audience, if you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. In addition to our presenters, we'd like to welcome the following panelists: Dr. Wenlei Zhang, Senior Advisor for Innovation and Strategic Outreach, Office of Research and Standards, OGD, and Dr. Xiaozhen Zhang, Deputy Division Director, Division of Bioequivalence 2, Office of Bioequivalence and OGD. We have some questions coming in right now. And the first group of questions will be directed to Dr. Sarah Ibrahim. And here's the first question for Dr. Ibrahim. Who are the 7 regulatory members of the generic drug cluster?
For the question. The current members are, of course, FDA, and then the regulatory authorities that represent the EU, which is EMA. We have United Kingdom, Swissmedic, Health Canada, and representatives from Israel, and the latest addition was Australia, TGA.
Thank you for responding to that question. We've got a couple more questions for Dr. Ibrahim, and here's the next question. How can other regulators collaborate with OGD outside the members of the generic drugs cluster?
Thank you for the question. As I mentioned, the generic drug cluster consists of the 7 regulatory agencies that, based on the confidentiality commitments, have a broader range of topics that can be discussed. However, the Global Affairs Program and a number of the international offices within the centers, as well as the Office of Global Policy and Strategy, have numerous methods by which we interact and share technical information with other agencies. And then, of course, we have our collaborations through external organizations such as IPRP and ICH, and those allow for venues with further engagement. The best answer would be, if you don't know, then reach out to the Office of Global Policy and Strategy, and they will assist in navigating to the level of collaboration that we are able to have with those regulatory agencies.
Thank you for responding to that question. We do have one more question for Dr. Ibrahim. And here's the question. How frequent is the international collaboration and engagement outside the scope of the generic drug cluster?
Thank you for the question. I think we've seen through all the presentations that our international engagements span across a number of engagements. And as I mentioned, whether it's IPRP, GBHI, ICH, and of course the cluster collaborations, the parallel scientific advice, I would say that our international engagements are on a daily basis. In fact, in a recent poll, when we asked the public, are you aware of how often our engagements are with EMA? The reality is it is daily. So there are significant international engagements and collaborations amongst our regulatory agencies.
Thank you for responding to that question. Moving on to our next panelist, we've got a few questions that came in for Dr. Nullifar Tample. And here's the first question for Dr. Tample. Will the guidance document cover all dosage forms? As an example, injectable emulsions.
Hi, Ray. Thank you for the question. The scope of this M13 is limited to orally administered IR products, which are solid oral dosage forms, and which are also designed to deliver the drug to the systemic circulation. So it would include dosage forms like the tablets, capsules, the granules, powders for oral suspensions. It does not cover the injectable emulsions. But also at the same time, the principles for other dosage forms, such as for the rectal or inhalation or the nasal products, the principles of the in the M13 may also apply to these non-oral dosage products in case the action is supposed to be for an immediate release and also meant for systemic exposure. Thank you.
Thank you for responding to that question. We've got a few more questions for Dr. Tample, and here's the next question. You mentioned the FDA guidance on BE studies with PK endpoints for drugs submitted under an ANDA will be updated. Why is it necessary?
Sure. So FDA will move towards adopting the ICH guideline and issuing it as the FDA guidance for the industry once the M13 is adopted by the regulatory members of the ICH assembly. When this occurs, we recognize that the scope and the subject matter of the current PK endpoint guidance for the ANDAs will not entirely be consistent with the M13A guidance. And this can create confusion for the industry because applicants may question whether they need to follow the FDA's PSG or the general PK guidance or the M13A. And also remember, this FDA's PK guidance covers both the MR as well as the IR products. So FDA will not be able to just simply decommission or withdraw the existing guidance. And therefore, FDA will need to make the necessary updates to the guidance to eliminate the discrepancies between the recommendations for the IR products, between the two guidance documents, the ICH and the FDA guidance documents. And also, we want to retain the relevant portions of the MR products, as well as certain aspects of the IR products that don't fall into the scope of the M13A. So that's why we would need to revise that guidance. And for the same reason, FDA will also need to update some of the PSGs. Thank you.
Thank you for responding to that question. We do have another question, another two questions for you, Dr. Tample. And here's the next question. Can you elaborate on the research with the Bayesian approach for Fed BE studies?
Okay. Sure. Thank you for raising that question. The Bayesian statistics, it's a particular approach for applying the probability to a statistical problem. So this approach starts with a summary of our prior beliefs, and these beliefs are based on relevant available information. So for this particular project, our idea is to assess the feasibility of performing a second study, which generally would be a Fed study with a smaller sample size, by borrowing the results of the first study as informative prior using the Bayesian dynamic borrowing approaches. And in our case here, the first study would often be the fasting BE study. So we are seeing this approach being used in pediatric trials, and we want to investigate its applicability for the Fed BE studies. And we hope that this approach may offer some advantage of reducing the human exposure to the study drugs and reduce the burden on the generic drug development programs, as well as it would help us to improve the efficiency of our drug approval process. Thank you.
Thank you for responding to that question. We do have one more question for Dr. Tample, and here is the question. How could high-risk products be determined, and are there some guiding rules for that?
Thank you for the question. I think although it's not directly related to the scope of today's talk, I'll address it because this question may be on many people's minds. So first off, I would want to emphasize that the high risk here, the term high risk, is not referring to any specific risk to the patients. It is referring to the risk of incorrectly concluding bioequivalence of a product if it is tested only under one condition. Generally, that would be the fasting condition. So there are certain drug products containing low solubility drug substances which may be formulated using specialized technology or manufacturing methods to enhance their solubility or to optimize the food effect. And M13 mentions some of these examples, like the microemulsions, the solid amorphous solid dispersions, lipid-based formulations, and nanotechnology, where the food can have a differential formulation-related effect. And in that case, it may not be appropriate for us to extrapolate the results of a fasting BE study to conclude bioequivalence under fed conditions. And here we may have some less experience and knowledge, and there may be more challenges that are involved with this type of products. And then there are other examples which are also mentioned in the M13A, such as the differences in the particle size of the API between the test and the reference products, or certain excipients that can affect the absorption profile of the drug substance differently under the fasting and fed conditions. So I think the M13A nicely covers the guiding principles for the high-risk products, along with the examples that also I just mentioned. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, we've got a few questions for Dr. Lei Zhan. And here's the first question. What would be the fees for the pilot PSA?
Thank you, Ray, for the question. So for the PSA, as the PSA involve both EMA and FDA. And from EMA side, they follow more like the existing scientific advice process. And for the FDA is more like our GDUFA meeting process. As you may see in the principal document, there will be the EMA fee, which whatever the fee applicable for the scientific advice process, it will be applicable to PSA meeting. But from the FDA side, there will be no fee to participate in the PSA program. So if you're not sure what that fee is for the EMA side, please contact EMA directly for that question. Thank you. And also, Ray, I did do a poll in my presentation. I think maybe can we take a few moments to just quickly review the poll to everyone. Thank you.
Thank you for responding to that question. We do have a few more questions. You certainly can review the poll. And here's the next question.
So do I review the poll now?
Please,
I saw the poll was, thank you. So the first question, I want to know how many people before this webinar or SBI workshop heard about the program. It seems that we have about 50% people know about, which is great. But for another half, people who never heard about or not sure. So hopefully you learned from my presentation what this FDA EMA parallel scientific advice pilot program is, what's the goal, what's the benefit. Next question.
Thank you for that review, Doc.
Oh, thank you. Okay, next question. I just want to see for those who are aware of the pilot program, are you considering your company to participating? It seems like about half people that their company do not develop complex generics or hybrid products, so they may be out of scope. However, we do have at least 15% consider will be participating and also have another 28% thinking about participating. So hopefully for those who are interested, today's presentation will help you to navigate through this new pilot program and consider using it to facilitate your drug development program. Thank you.
Thank you for that review, Dr. Zhan, and we'll go on to the next question, and here is the question. As per the pilot PSA guidance, the information regarding RPI, Research Product Identifier, is required to be included in the meeting request. Can you please elaborate more on the RPI?
Okay, thank you for the question. First, I want to clarify that it is not a PSA guidance. It's more like a general principle document to lay out the process for this pilot program. And in the process, the person did notice there's a research product identifier is required, but that's a requirement from the EMA side, because this is really their identifier for the active substances. And also they require people to register RPIs through their IRS platform for this number. So I would like to, since this is the EMA requirement, I would refer the person to the EMA guidance on the IPI for further information. Thank you.
Thank you, Dr. Zahn, for answering that question. We do have a couple more questions, and here's the next question. What would be the timeline for submission of the meeting package for validation after the meeting request is accepted?
Yeah, thank you for the question. And this is really depend on how quickly the applicant to get the full package ready. And also this is mainly an optional step from EMA side. You can submit to EMA to review your draft full package for validation before you submit the full meeting package. So typically I learned from EMA is the company can take as short as 2 weeks to like 2 months or even longer to submit that full package. So this is really the timeline controlled by the applicant. Thank you.
Thank you for responding to that question. We just have one more question for Dr. Zahn in this round, and here's the question. In past experience, FDA did not allow us to use the EURLD for a BA study of a complex generic product, even though the EURLD and US RLD is essentially the same. Will this PSA program help the applicant to reduce duplicative BE studies?
Yeah, thank you for the question. I cannot comment on what this person who gave the question, the experience is. I cannot comment on that, but through my presentation, you do see this PSA program. The goal is to help the applicant to reduce the duplicate BE study. If you found there's a challenging in your case, please submit a package and pose a question to both EMA and FDA, and we can see whether there's a path forward to design a study that can satisfy both regulatory agencies' needs or requirement. Thank you.
Thank you for answering that group of questions. Moving on to our next panelist, we have a few questions that came in for Dr. Brian Folian, and here's the first question for Dr. Folian. Understanding that CDER's Office of Study Integrity and Surveillance, or OSIS, conducts many remote regulatory assessments, do you see potential data integrity concerns as being suitable for examination via an RRA?
Thank you very much. That's a really interesting question and very timely. I think, as I discussed in the presentation, when we look at data integrity, right, we're considering, whether or not the data in front of the agency submitted is complete, consistent, accurate, and reliable. So for purposes of a remote regulatory assessment or an inspection, the crux there is really determining what we can actually see, what the actual issue is at hand that might be raising the data integrity concern. A lot of times we might identify something during an RRA that would actually be indicative of some concern with regard to the study sites, study conduct, and the data they produce via that actual conduct, right? So if there's something submitted to the agency and we go to a firm and we're remotely interacting with them and we notice that their records don't support something that's submitted to the agency, you start to open questions. So whether or not that's via remote or on site, you're doing the same thing, somewhat that antenna goes up, right? You start actually looking into it and you start, doing a deep dive and trying to figure out what you can see. There is some intricacy to that in a remote setting, obviously, but, over the past 3 years and even prior to that, we've been learning a lot about how these interactions can occur and, getting more comfortable with, the technology and how we engage. So whether or not we have a data integrity signal identified, prior to determining what interaction is appropriate with a firm, or if we're in the midst of an RRA with an entity, it's all case specific in terms of whether or not, we can start digging into that and seeing what the entity has, asking questions, reviewing the data on site and the data that can be shown when we're remotely looking at with the firm, what their study files look like, what's been submitted. So generally, there is the potential. You can never say carte blanche that everything can be looked at comparably with the remote versus inspection, because they are, as everyone knows, and different things. They're both fit for purpose tools. So we're still continuing to evaluate and work with RAs to determine the extent to which they can support the agency's, inspection-related activities, so we can make sure that the products we're approving, have reliable data to support them. Thank you very much.
Thank you for responding to that question. We do have a couple more questions for Brian Folin, and there's one for Dr. Folin, right? The collaboration and complexity of FDA and international regulatory is really interesting. So, to the extent possible, can you provide more discussion on how, practically speaking, different outcomes may be reached?
Thank you again for this question. I think as Dr. Ibrahim's talk earlier in the presentation, as well as mine, it highlights the uniqueness, the shared interest in trying to reach that harmonization and commonality in terms of approaches, but also recognizing where we have differences in the way our regulatory oversight is effectuated. So it's an interesting period where you'd have, you would see a notice of concern from the World Health Organization or an action being taken by the European Medical Association. And what does FDA do with seeing in through our collaborative efforts with those entities? It's a really interesting dynamic scenario, right? Because as we've heard in some of the questions, it's interesting where people think that there's duplicativeness or the harmonization is possible, recognizing even if that is the shared goal, the underlying basis for why WHO might find an action supported might not necessarily be the same issue that the FDA may have in front of us, right? There's always differences in terms of study conduct that's submitted to WHO or study conduct that's submitted in support of data that's submitted to FDA. So we always have to do that very specific consideration of the information in front of us that can lead to different outcomes. You might have comparable drug products being examined, but overall the reality is that we have to do our due diligence to consider the information in front of each of the regulatory agencies in effectuating our regulatory oversight, which shouldn't be construed to say that we aren't supportive or otherwise constantly considering what each of us are doing and trying to the extent possible to be in the same area, but that isn't always going to be the case. So further work, as Sarah mentioned, is ongoing endeavors across the universe of what FDA does. We continue to collaborate and seeking that commonality where we can. Thank you.
Thank you for responding to those questions. Moving on to our next panelist, we do have a question came in for Dr. Wenli Jiang. And here's the question for Dr. Jiang. I know there is an International Pharmaceutical Regulator Program Nanomedicine Working Group. And this working group, is there any activity ongoing related to generic products?
Thank you, Ray, for this question. Actually, the simple answer is yes. I can elaborate a little bit more. The International Pharmaceutical Regulatory Program Nanomedicine Working Group, abbreviated as IPRP-NWG, works on the exchange of non-confidential information on nanomedicines among IPRP Nanomedicine Working Group members and the regulatory harmonization on nanomedicines. So this working group discussed topics related to innovator drug, generic drug, or hybrid products involving nanotechnologies. Currently, there are 15 regulatory members in IPRP Nanomedicine Working Group. In 2020, the working group conducted a survey on liposome and the lipid complex products among member agencies as well as non-regulatory stakeholders to provide a comprehensive analysis of the regulatory landscape for liposomal products to identify the needs of both research and standards development in order to enhance the potential for harmonization of regulatory requirements. Actually, based on the survey response by regulatory agency representatives, the majority of respondents indicated that they had no review or approval experiences with any follow-on or generic liposomal products. Several agencies reported they had approved generic liposomal products, but only limited to like pegylated liposomal doxorubicin hydrochloride and liposomal amphotericin B injection. And most agencies consider it is challenging to assess generic or follow-on liposomal products. For example, they do have questions. What are the appropriate equivalence criteria to compare innovator and the generic liposomal products? For non-regulatory stakeholders, for example, we conducted a survey among academic institutions, pharmaceutical industry, trade association representatives. And based on the response, the call for alignment of critical quality attributes for liposomal products among different regulatory agencies. And also they recommend guidance development to address the evaluation of generic or follow-on liposomal products. Considering these survey responses, the working group had quite some activities going on, like we have information exchange on scientific and regulatory considerations for generic and follow-on liposomal products. In the past 2 years, actually, we have organized webinar presentations given by the agency representatives who had experiences in reviewing generic liposomal products to IPRP Nanomedicine Working Group member agency scientists. Actually, they shared their agency review criteria and also the rationales supporting such review criteria and the review experiences. So the agency, which hasn't reviewed the generic or follow-on liposomal products, can gain experiences in attending these webinar presentations. In addition, some similarity and difference among different agencies were identified during the discussion. Hopefully, some scientific consensus can be reached among IPRP Nanomedicine Working Group member agencies regarding any specific generic liposomal product. Also, a number of liposome standards were developed by ISO and ASTM. So these standards were very useful for both innovator and generic applicants working on liposomal product development. IPRP Nanomedicine Working Group have been helping disseminate these standards and provide training to member agencies. Yeah, that's about it. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we do have a question came in for Dr. Xiaojun Zhang. And here is the question for Dr. Zhang. For oral extended release products intended for administration only under fasting conditions, can the agency consider waiving the requirement for a fed study?
Thank you for the question. ICH M13A guideline applies to immediate release solid dosage form. Therefore, oral extended release product is not in the scope of M13 guideline. The harmonization on modified release product will occur at a later time.
Thank you for responding to that question. We have just under a minute left. We're going to try to squeeze one last question in, and this will be directed to Dr. Sarah Ibrahim. And here is the question. Is the generic drug cluster an appropriate place for discussion of a single global comparator's development?
Thank you for the question. The generic drug cluster is a confidential forum. There really is no topic that we do not raise in the cluster if there's a public benefit for our citizens. And most of our engagements are on scientific engagement. So there's no
true limit to the level of scientific dialogue that is ongoing in the cluster, so long as there is a benefit and access for our patients.
Thank you for responding to that question, and we have run out of time for questions in this final Q&A session. A huge thank you to our speakers and panelists over the last 2 days for answering numerous questions that came in from our audience. Before we provide some housekeeping reminders, we'd like to welcome back Dr. Lineberger to provide some closing remarks for advancing generic drug development. Welcome, Dr. Lineberger.
Thank you, Ray. This was a fantastic workshop. First, I want to thank the SBIA team for all of the logistics, the organizing committee for putting together the program, and finally, the speakers and the panelists for providing the fantastic comment. All of those people had to work together to have a successful event. At this workshop, you heard updates on complex science that can support the development of complex generics products. You got practical advice from FDA staff who assess your applications as to what they look for when reviewing some of these novel approaches. This is a very unique and practical insight for those of you in the industry and a perspective on what happens to your application and how FDA staff look at it. You saw examples of complex generic products that were approved, and you gained some insight into some upcoming regulatory changes or new programs such as our model-informed evidence and our global harmonization. But now you know what's possible in all of these. Let's talk about what some of the things you can do with the information you've learned about this or the insight that you saw from today's workshop. The goal of this is, in one sense, to bring generic competition to every product, even those complex products that don't currently have any generic competition. And from the industry perspective, these are huge opportunities for you and your organizations to contribute to public health, but also to, for the business opportunities as well as being the first company to provide these opportunities. And today, during the Q&A sessions, we tried to answer as many questions as possible. But we know that you have more questions. We also know that you have many questions that are product specific and specific to your particular development programs. We can't answer all of those at this forum. And answers to these questions will help fill in the gaps for your development programs. And so what can you do? The best thing you can do is to use, for complex generics, is to use the pre-NDA meeting process to get the scientific advice before submission. This is exceptionally valuable, right? Because we all want the review process to be as efficient as possible, to get timely evaluation and approval of new generic products. But the way to do this is to get things right the first time. And the way to do that is to make sure that as many of the scientific issues are resolved through the pre-application process as you can. So use the product development meetings. During GDUFA 3, we have a new pre-submission meeting for complex products. We encourage people to use that meeting process as well to help share insight into what you're doing with FDA and get appropriate feedback on that, as well as to use the control correspondence as well. Today's meeting, you also saw an illustration of FDA's commitment to more meeting opportunities. You heard for the first time about our model-informed evidence pilot program. This is an expansion to the type of questions that you can bring into the meeting process. So it covers modeling and simulation related questions that might be applicable to more than one application or to non-complex products where you're supporting some novel approach. So again, our commitment is, we recognize, and the theme of this workshop is moving, how do you use the science to move products through the review process and make them available to the public? And the meetings and the product-specific engagements are such a key part of that, is that we hope that what you'll take away from this are the opportunities for developing these complex generics and the places you can go to interact with FDA to get feedback on the scientific issues facing these development products. And again, just to close out, we have a shared goal to make generic versions of complex products available. And this workshop is a very important part of that collaborative effort. So you can really see across 2 days of content a lot of the different areas of opportunity across many different categories. And so we hope this workshop has been very helpful to you and helps you develop complex generic products and points you to many different ways that you can interact with the agency to have scientific discussions about those development programs. And so with that, I want to turn it back to Ray Ford and the SBI staff so they can close out the housekeeping